Okay, so this is the second video for cell membranes. At the end of this slideshow, you should be able to compare and contrast endocytosis with exocytosis and give examples. Compare and contrast passive and active transport, give examples. Different forms of passive transport, including simple diffusion, facilitated diffusion, and osmosis. And can you explain how the ability of the plasma membrane to be selectively permeable maintains homeostasis within a cell? So, here we go again. Here's another picture of our cell membrane, the phospholipid bilayer. Now, we talked about this a little bit last time, but there are two types of transport through the membrane. There's passive transport, which does not require energy, and there's active transport, which does require energy in order to get movement through the membrane. Passive transport is often called diffusion and does not require energy. Molecules are going to move from an area of high concentration to low concentration. You've experienced something like this happen a lot in your life. So, for example, when you mix in lemonade mix into a bottle of water, for example, or a cup of water, that lemonade mix diffuses throughout the whole thing. This is an example of passive transport if it's going through a membrane. So, for example, here we have our layer and our phospholipid bilayer, and it is the example that you would want to write down is it plays a crucial role in the import of resources for reactions and the export of wastes. So diffusion is the passive movement of particles from an area of high concentration to an area of low concentration. Also another thing that you guys have already experienced. This results from random collisions between particles and does not require any energy. So once again, diffusion is going to help with the import of resources, export of wastes, does not require energy, moves from an area of high concentration to an area of low concentration. Here's another sample. So another thing we have is called simple diffusion. Simple diffusion occurs when particles pass through phospholipids and diffuse across the membrane without the aid of a protein. So it is only applicable to molecules that are permeable to the membrane. So right here we have simple diffusion because things are going straight through the membrane, still from an area of high concentration to low concentration. Ions and molecules that cannot pass through the membrane like that through simple diffusion go through facilitated diffusion, which does require energy and uses some sort of protein channel. So our protein channels are going to be different in their structure. Some are going to be more negative, some are going to be positive. So in this situation, you can see that opposites attract here. So our positive charged particles are able to pass through this negatively charged channel, whereas in this one, our negatively charged particles can go through our positively charged channel. A really, really important example that you are going to need to know for this one in biology for facilitated diffusion is what's called the sodium-potassium pump. So that's an image here. And sodium and potassium are going to be passing through different protein channels and helps with neuron signaling. So in a sodium-potassium pump, what's going to happen is we're going to have our potassium exit the cell and create a gradient here. And through that gradient, sodium is going to enter back into the cell, and this is a great example of facilitated diffusion. Osmosis is the passive movement of water across the semipermeable membrane. We already know this one. It's going to move from an area of high concentration to an area of low concentration. Hypertonic is an area with higher solute concentration, and a hypotonic is an area with lower solute concentration. So, for example, in our image here, these little dots are representing our particles that are in the, whatever this is, probably water or something. We could consider the cell to be hypotonic because it has a lower solute concentration. And this environment out here is going to be hypertonic. We talked about this in class with the example of the salt water or the ocean water and our human cells. Because the cell is hypotonic and the environment is hypertonic, the net movement of water is going to be out of the cell because we are trying to go from an area of high concentration of water to an area of lower concentration. Okay, water movement occurs until the solute concentration is equal, so that's isotonic. So isotonic means that the solute concentration on the outside is the exact same as the solute concentration on the inside. Note that there's still going to be water movement both in and out of the cell, but it's going to be more equal. So in a hypotonic solution, what's going to happen is water is going to enter into the cell, and a possibility is that that cell becomes lysed, which is basically where it's going to explode. In an isotonic solution, water will move in and out. And then in a hypertonic solution, water is going to move just out of the cell and cause the cell to become shriveled. We have a very similar process in plant cells. However, the one thing that is different is that this is actually going to be hypotonic solutions. This is actually going to be normal conditions. Plant cells have the capability to not become lysed the way that animal cells would. So that is a really important feature that they have. In an isotonic solution, water is going to be moving in and out, but that's not going to be normal. Here's another image of our hypertonic, isotonic, and hypotonic cells. Okay, so this is pretty interesting because most tissue samples are stored in normal saline, which is a sodium chloride solution, and it has the same osmolarity, so it does not cause damage. So that's why a lot of times in order to rinse out wounds or mycin-damaged areas, eye drops are going to be a saline solution, is because we don't want it to have either too much salt or not enough salt in it to cause the cells to be in either a hypertonic or hypotonic condition or environment. So we want it to be isotonic so that there's no permanent damage. So that is why a lot of times with medical procedures they're going to use a saline solution. All right, moving into active transport. Active transport is a form of transport that requires the use of free energy, which is going to be in the form of ATP. And it moves particles from areas of low concentration to areas of high concentration, so against the normal gradient. So because it's going against the normal gradient, that's why we require energy. So as you can see in this top one, we have an example of simple diffusion, where things are going just straight through the membrane. We have facilitated diffusion. Both of these are types of passive transport. Facilitated diffusion is where things are going through a protein channel but do not require energy. They're both going from an area of high concentration to an area of low concentration. But over here we have an example of active transport in which our particles are going from an area of low concentration to high concentration. In order for this to occur, we have to have the input of ATP for energy. Okay, so one type of active transport is through protein pumps, which are proteins embedded in the membrane that are able to push molecules against their gradient. So you can see here our hydrogen plus molecules are being pushed outside of the membrane. All right, so we do have in the cells, we have something called vesicles, which are used to transport materials um, throughout the cytoplasm without mixing anything. So it kind of creates this little barrier here. Our vesicles are also going to be made up of a similar or the same phospholipid bilayer, and they're actually able to move in and out of the cell. So here's an example. This is showing exocytosis. So you have our vesicle here that has the phospholipid bilayer and whatever particles are on the inside. And what's going to happen is that it's actually, we're showing it the opposite way. Hence the arrows. So this is an example of actually endocytosis. I didn't see this arrow at first. Endocytosis, because all these particles are coming into the area on the of the phospholipid bilayer, and the bilayer is actually going to close off here. You can see it's starting to close, and it's actually going to form a vesicle. This vesicle is going to help transport these particles to wherever it needs to go within the cell. Now this is considered endocytosis. Endo, once again, meaning that like within or internal. Now this could also be, like I was explaining before, exocytosis, if these arrows were facing the opposite way, where it's going to go ahead and bring them out of the cell. The process of bringing materials into the cell is called endocytosis. Endocytosis is an active process, so it requires ATP in order for this to occur. So there are three main types of endocytosis. The first one is phagocytosis, which is going to bring in any sort of large cells and particles. So pretty much anything that is solid is going to be going through what's called phagocytosis. Penocytosis is only going to bring in water and other liquids. And then finally we have receptor-mediated endocytosis. And receptor-mediated endocytosis is where there is some sort of receptor on the outside of the phospholipid bilayer, and on the inside there's going to be these proteins that are going to end up coating the outside of the vesicle. So once that receptor has been signaled, it's going to close off and transport whatever it received through that vesicle. But it's important to know that there's three different types of endocytosis, and understanding what each of those are, types of endocytosis do. So exocytosis is the opposite of endocytosis, in which things are going to exit out of the cell. So what's going to happen is the Golgi apparatus is going to often package materials up into small vesicles. You can see that process occurring here. And those vesicles are going to travel to our phospholipid bilayer and release whatever the contents were out into the environment. Because of the membrane's fluidity, the membrane of the vesicle is able to fuse with the cell's membrane. So we talked about this a little bit in the last video. The importance of a fluid mosaic model is because endocytosis and exocytosis occur, well, part of the importance is because these two processes occur so easily. So know that if there's anything asking about why membrane being fluid is important, well, part of it is because vesicles can form, can fuse, or can break off quite easily from the plasma membrane, which that will help increase the amount of cell membrane overall. And then obviously that helps for cell growth because we're increasing our surface area to volume ratio. Now, this process of endocytosis is really crucial, and exocytosis is crucial for maintaining cell homeostasis. So homeostasis is just maintaining those internal conditions. So having these two processes allow, well, not only endocytosis, exocytosis, but that and active transport and passive is really important in maintaining those internal conditions, because you're allowing things to leave the cell that should not be in the cell, and bringing things into the cell that you are lacking. Here's another image of endocytosis versus exocytosis. This top one is obviously an example of exocytosis because arrows are coming up this way, releasing it into the environment. Endocytosis is the formation of those vesicles. Transport with vesicles allows cells to take in large particles. Obviously, endocytosis digest them and then excrete the waste. And that's pretty much it. So just as a review here, and go back up to these. Make sure we got them all answered. Compare and contrast endocytosis and exocytosis. Bringing things into the cell, taking things out of the cell. Passive and active transport we covered. Simple diffusion, facilitated diffusion, and osmosis. And then can you explain the ability of the plasma membrane to be selectively permeable, maintains homeostasis. So what I would recommend doing at this point is now kind of going through and finding if you can see specific examples of endocytosis and exocytosis. There's going to be a ton, and same with passive and active transport. We covered more of the examples for this, but I would look up specific things for endocytosis and exocytosis.